Well, ladies and gentlemen, distinguished colleagues, there are lots of participants today with us online, so we must start, roughly 2,500. So this is our second plenary session of the fourth St. Petersburg Innovation Medical Forum. Today we are going to consider issues of individualized medicine, and this is a large-scale event on the basis of the Innovation Center for Personalized Medicine based on the Almazov Center. I would like to give the floor to Andrei Rodskoy, Peter the Great University rector. Distinguished participants of the fourth Petersburg Innovation Medical Forum, it is a great honor for me to deliver the welcoming address at the Almazov Forum, as we call it in Russia and in St. Petersburg, because this forum, on the basis of the Almazov Center, considers cutting-edge advances in medicine, biology. In 2015, the Almazov Center led the initiative on translation medicine, which emerged into a cluster that has brought together different stakeholders, including the Peter the Great Polytechnical University and many other stakeholders. This cluster covers highly critical topics related to national healthcare, which is the priority for our government and all the governmental institutions today. I would like to note that Peter the Great Polytechnical University has broad capacities in performing research. We have high processing, big data, in bioinformatics, in AI, digital twins, and many other complicated systems and technologies. We continuously contribute humbly into the most relevant advanced research in medicine, starting from oncology to the plague of the 21st century, coronavirus, that is still with us. As for coronavirus, you know that Evgeny Shlekhto, I wanted to congratulate him with the new title of an honored citizen of St. Petersburg. And you know that last year, Evgeny Shlekhto was elected honored citizen of St. Petersburg, and he now leads the work group on coronavirus proliferation and countering coronavirus proliferation and expansion. So last year was indeed a tragic one, and regrettably we were unable to host a number of events due to the lockdown. And I would like to note with appreciation that this hybrid model has become the cornerstone format for conferences and events, allowing to analyze statistic data and refer to specific developmental scenario for a particular region, step up very accurate governmental decisions. Let's consider the Republic of Komi, despite the forecasts that we cannot relieve the lockdown when the incidence rate went down, we were able to suggest mathematical evidence that there will be a huge spread of disease and that the incidence would skyrocket. And this is the scenario that happened indeed. So the lockdown was reinstituted immediately. It is thanks to us that we were able to build relevant forecasts. In the same manner, we cooperated with Professor Schlechter in St. Petersburg. On a daily basis, we produced recommendations to the governor of St. Petersburg on how to counteract and how many beds would be required for a particular day and what logistic decisions would be most feasible. So there is a lot of theory about that, but nevertheless, these efforts contributed largely to a safer scenario, and these allowed us to avoid a most unfavorable scenario for St. Petersburg and northwestern region. On top of that, in the meantime, within the framework of this cluster, with the cooperation of the flu research center that is developing mucosal vaccines, and today actually we are having professor and director of the Biomedical Technologies Institute, who works in close cooperation with us. I would like to note this particular achievement of the Almazov Center and our university. Those are the winning positions in a specific competition. Today, the Polytechnical University and the Almazov University are the coordinators of this consortium. One of the topics is digital modeling and forecasting in medical and biological systems. Of course, naturally, we cooperate closely with scientists from the Almazov Center, and this is closely related to digital platforms for RNA and vector vaccines, R&D, development of immune and biological drugs, and different systems to promote vaccinations. Within the framework of this forum, speakers looked into highly crucial issues of contemporary medicine. Actually, I believe that the entire spectrum from hematology to such diseases of our century as oncology, cancer.
And this is one of the themes that is of great concern to everyone. Of course, cardiovascular diseases are quite important for the Almazov Center, but I know that they have expanded their activity beyond that trend, and they are, so to say, a catch-all center of medical research, advanced technologies, being a rector of one of the largest high schools of Russia. I would like to acknowledge the Almazov Center and its administration creation of a unique medical university, which was set up several years ago, and I will explain why medical high schools should be chamber schools, and treatment and learning should be personal, and all the students should be high-grade students. I will never go to a student who has poor score. That rule should be implemented, at least in Russia. I would also like to note that we're also using our supercomputer center, which is among the 5 top computer centers in Russia. We're also creating the intelligent assistance and the systems for prediction using the processing of big data, and we have developed special software under the leadership of Professor Lev Utkin that is an intellectual system of diagnosis. Using one of the examples, these are lung tumors together with the City Oncological Center, and that software that can be installed in each modern computer within 20 seconds would analyze a CT of the lungs of the patient, compare them with hundreds of thousands of similar cases included into the software, which we could digitize. I mean, we digitize the data on the disease, and immediately a resolution will be passed as the pathology and statistics and the possibilities of coincidence with the diseases. I'm not a doctor, but I have been said the most experienced oncologists say that even proceeding from the CT of the patient, they say that the probability of diagnosing an oncological disease at an early stage is only 15 to 17%. I just proceed from the data given to me by my colleagues who are doctors using our methods that has been tested for about 2 years, and I think it will be applied to a broader range of research possibilities. And there the probabilities from 90 to 94%, I mean the precision of diagnosing the disease, and it is quite clear. I would like to focus on the development, our joint development with Schwaber concern. Schwaber is also a strategic partner of Almazov Center and our strategic partner. This is the development of unique for Russia new device diatom that is under clinical trials now, and its batch production is being prepared at Novosibirsk instrument making facility. This is a very powerful ultrasonic equipment where the ultrasonic device and the ultrasonic robotized gun is within the same system that is for mammology, for finding malignant and benign neoplasms, soft tissues that can be used in mammology using ultrasonic procedure, and that software makes it possible to determine the depth, the size, and the possibility of using the ultrasonic gun on that neoplasm, and that gun would result in dissociation of that neoplasm in human organism. Such equipment is also manufactured in France. But what have we done? We combine science and technology and robotics as one of the spheres of high production computation. Unlike the French device, it looks as an installation, as a CT. And here that is just a robot that determines everything immediately, and the patient can be seated and be subject to the necessary treatment under comfortable conditions, and it will be he or she will be cured at early stages for certain diseases related with neoplasms. I think this is an outstanding work. Currently, we are working with the experts in the sphere of prostate diseases, urologists. We're also working with the Hudson Institute. We have been discussing these issues, and I believe that we're going to, on the basis of these effects, we're going to develop the devices that would make it possible without any serious impact on human organism to treat the diseases. As you know, nature and science and medicine are working jointly, but I can state with pride that we also make not only the scientific contribution to combating oncological diseases such as leukosis at children's centers. Recently, we signed an agreement with the National Registry of Bone Marrow, and hundreds of our youth have become involved, and they will be donors. And the next section will be held on the 1st of June, on the Day of Protection of Children. Probably I'm saying too much words, and now I would like to come to the conclusion and tell you that the work at that forum will be a new stage of development of medicine of the global level, and which at present is a combination of science, technology, and medical science. And the main thing that I ask you to have a strict civil position. You have to acquire new technologies and knowledge in the sphere of healthcare, because our life, to a major extent, depends on your mastership, your literacy, and exchange your experience. Golden hands and golden heads, which I wish you. Thank you. Thank you very much, Andrei Ivanovich, for the opening of our plenary session. I would like to tell you that we place major emphasis on the development of our cluster translation medicine, and we hope that we'll be able to find additional possibilities of additional interaction. Allow me to give the floor for the brief presentation, welcome address to our third co-chairman, Sergei Bakulev, rector of Lesgov University. A very important component of our cluster as well. Dear friends, distinguished colleagues, I'm thankful for the honor to represent the oldest sports school, not only in Russia but in Europe, the National State University of Physical Culture, Sport and Health, named after Lesgov. This year we celebrate our 125th anniversary. I won't discuss our institute. I will discuss the cluster. Health is prioritized not only in Almazov Center, not only in our cluster, but at our university as well. The only difference is that Almazov Center cures people, and we try to prevent the disease using locomotor activity. Actually, we also participate in the work of Almazov Center, and recently, educational activities started here. They train doctors using modern laboratory equipment and practical skills. We also have the honor to take part in training. We provide our competencies so that people and experts and doctors who are graduated here will become also sports doctors. I'm ready to participate in such fora, because that is really an indicator of the efficiency of the work of Almazov Center, that demonstrates the efficient operation of our cluster, and it is pleasant that our experts take part in the activities of Almazov Center. Thank you. Thank you, dear colleagues. Now we pass on to the main program. It's with great pleasure that I give the first floor for the plenary presentation to Dmitry Alexeyevich Sychov. Unfortunately, he couldn't come here, though he had to attend in person. He will use Zoom to participate here. Dmitry Alexeyevich is a person who is a pioneer of personalized medicine and pharmacogenetics in the Russian Federation, and we are eagerly looking forward to listening to his presentation. This is the first presentation that he makes at our forum. Please, Dmitry Alexeyevich, take the floor. Thank you very much, Alexander Legana, dear panelists, dear colleagues. Thank you for making the presentation. And at the beginning of one's presentation, I would like to graduate St. Petersburg with the anniversary of St. Petersburg. I would like to thank Almazov Center and Evgeny Vladimirovich in person for the opportunity to deliver a presentation, the plenary session on personalized medicine in terms of clinical pharmacologies. I would like to thank all the colleagues for cooperation between Almazov Center and the Academy, director of which I am, the Russian Medical Academy of Vocational Education. I would like to start my presentation with a statement worded 60 years ago by a person who worked at our academy, then academician Watson, who 60 years ago said that we're far away from personalization and 3 times a day, everything is given to everyone except laxative and snow-causing devices. There are some drugs that can be administered once a week or 2 times a week or once a half a year, but we're far away from personalization. All our patients are different and they respond to the drugs in different way. The results of multi-center clinical trials give certain average results on the efficacy and safety of drugs. And these studies underlay the development of clinical recommendations of standards of medical assistance. We have to understand that the drug response at adverse reaction depends on many factors, but genetic factors might account for 50% of response, though other factors also affect the development of adverse reaction resistance, that is sex, age, inter-drug interaction, bad habits, physical activity. We have to realize that now we are essentially at the beginning of development of personalized approaches and medicine. The model of medicine that we have is a reactive model, the upper diagram. So you have clinical recommendation. You can see a set of drugs that prove to be efficient in large studies, and the doctor has to choose certain drugs and start treatment. He starts treatment, combines the drugs, increases the dose, decreases the dose until he reaches the objective. For instance, the figure of arterial pressure or cholesterol level. Then the risk is diminished and the complications will not be caused. We want the medicine not to be reactive, but personalized and precision. When we have specific biomarkers that predict the efficacy and safety, we select to the patient the drug precisely which would not cause any side effect, would be effective. This is an ideal model, and sometimes we even get some results due to it. That is the necessity of passing on to personalized medicine that is recorded in the strategic documents of the country. That is the scientific technological strategy of development of the Russian Federation, strategy of development of national health care under the concept of medicine. We said we talk about 3, 4, or even 10 medicines. All of us understand that there is the same, not P medicine, a certain methods that helps to select the patient. For him, the drug would be safe and efficient and will not cause any adverse effects. Why the knowledge of personalized, notion of personalized medicine is a biomarker? This is realized in the concept of our medicine developed in 2018. Biomarkers which interest clinical pharmacologists who are dealing with pharmacotherapy are the biomarkers which predict individual features of pharmacokinetic of the drug and the therapeutic concentration for the patient. That range will fix the lower range of side effect and the highest efficacy. Those biomarkers should reflect the individual features of this process: absorption, distribution, metabolism, and excretion, which function due to the work of special protein systems, enzymatic systems, transport systems of drugs. Our teachers long dreamed of personalization. Even the phrase by corresponding member of the Academy of Medical Science, Professor Larkin, well-known pharmacology that will start individualization of the treatment when we can control the fate of the drug in human body. And modern technologies that have been discussed are moving us toward real management, personalization. At our academy, we develop biomarkers. We predict the efficacy and safety of using a broad range of drugs in the therapeutical clinic. With Almazov Center, we also cover broad areas, and now there is a work on searching biomarkers for the efficacy and the safety using traces. Biomarkers that were developing pharmacogenetic biomarkers based on different PCR tests. Now we use the new generations. These are a priori biomarkers, and they can be. We can predict the response to the mark. This is pharmacotranscriptomics about plasma RNA and therapeutic diseases, particularly the expression, if it is related by expression of genes, and pharmacometabolomic biomarkers determining the concentration, metabolites, and drugs, and phenotyping based on the study of metabolites of the systems of transport and biotransformation of the drugs, and they are more costly, and that makes it possible against the background of treatment start correction. These are new methods, and I would like to show how we study and implement them in clinical practice. One of the methods is phenotyping of the system 6-beta-hydroxycodone. This is subfamily 3A that metabolizes a lot of drugs. We assess the activity based on concentration of cortisol, 6-beta-hydroxycodone in urine. Why? Because this branch of metabolism of cortisol is catalyzed by a CREAS system, 3A system, and developing this methodology, the activity of such material correlates with parameters of safety and efficacy. This is a clinical situation of oncological patients. We used benzodiazepine tranquilizer, diazepam. There was a correlation between the activity and safety and efficacy parameters based on psychometric skills and Oku school scale. I'll give all the examples based on the published foreign publications. Another work on the same biomarker activity of 3A is dedicated to the study of biomarker for predicting thrombosis of stents in patients with acute coronary system that get double anti-aggregate therapy. Another biomarker characterize the activity of another enzyme, but the 6 all the beta-adrenobrate. We have also developed a system to develop the activity on metabolite of pinoline. And there is a correlation between the activity of 2B6 and the parameters of another psychotropic, alpha-metazapine, in patients of oncological profile. We hope that another group of biomarkers, plasma microRNA, will be used. Their expressions related with expression of the gene of transport and biotransformation of the drugs. One of the first works was published where we found correlation between microRNA 27B and concentration of tranquilizer alprazolam in oncological patients. As for the patients with acute coronary syndrome, we have developed an original methodology of choice plasma microRNA for prediction laboratory resistance using not only clopidogrel, but also other drugs, for instance, pradagrel, antigrel in patients with acute coronary syndrome. We understand that these biomarkers, which I mentioned, pharmacotranscriptome and metabolomic and pharmacoproteomics and pharmacomicrobiome should work together so that this transition from personalized to precision medicine would be really realistic. Then we can precisely predict side effects or resistance to treatment. We understand that if we develop such approach, we have to use certain metabolomics, which now will be demonstrated. First, it is a choice in development of specific specific biomarkers, and on their basis, models and algorithms of personalization of the choice of drugs and the regimes of dosage. To do it, we have to understand what the medical problems would personalization solve. And the clinical pharmacologist has the relevant methodologies, pharmacoepidemiological studies. This is an active monitoring of side effects. That these are neuroleptics in such patients as adolescents with acute psychotic syndrome. On the basis of this study, we formed bioresource collection of patients with such side effects using neuroleptics, and we created a register of patients. And there are more than 20 different bioresource collections on side effects and resistance to treatment, more than 20 ethnic collection to assess the ethnic sensitivity based on the use of the drug. This is our real wealth, and using this collection, we can have associated studies. The studies on association between the chosen biomarkers and the relevant parameters of the efficacy, safety, pharmacokinetics, and pharmacodynamics. Pharmacogenetic of warfarin is quite important. Russia started it in St. Petersburg under the management of Professor Schwartz. First works were done, and it is metabolizes, it enzymes that affect the enzymes. We studied polymorphism of the genes, and we find association between the use of anticoagulant cognitive effect of warfarins and dose of warfarin and polymorphism data. Complication with other groups of drugs. We have used sequencing of the new generation of patients with bleeding against beta-cron Varexon. We find SNP that were not really described associated with these complications, and we include these data into the algorithm of personalization of using these drugs. And sometimes in clinical practice, we have long been used a certain drug, and it's used extensively, but there are some problems related to it. But we cannot select biomarkers because when the drug was developed, nothing was known about cytochromes, metabolization, transporters, how it is transported in human body. And here in this work, me and Fenazepam invented about 60 years ago in the USSR at Pharmacology Institute.
Currently, benzodiazepine is a tranquilizer that is associated with severe morbidity. And Civica program allowed us to see that cytochrome CYP3A and 319 are the most frequent ones, and we demonstrated that in vitro these cytochromes participate in phenazepam metabolism, and then we did a clinical trial to find correlations between the polymorphism of reactions and the gene disruption. And we published the results of our trials. After that, we had to see an associative trial to look at the correlation of these two biomarkers. And here you may see the polymorphism of CYP2019 and citalopram concentration in patients with depression. This polymorphism was associated with higher concentrations of CYP2019 and higher incidence of unfavorable responses. On top of that, we also look at the pharmacodynamics. For clopidogrel, for example, yes, CYP2019 can forecast resistance in patients with CAD. But when we did haplotypic analysis, we found out that specific combinations of alleles can forecast lab resistance better than just via gene phenotyping by CYP2019. And on top of that, we also tried to forecast the outcomes once we found the polymorphism of 2019 gene. We also found correlation with the death rate in patients with CAD who received treatment. Based on this research, and considering the cooperation of bioinformatics, we structured the forecasting models that allowed us to pack these models into the decision-making system. And these models are highly required by genetic doctors who allow doctors to develop a personalized approach to treat patients. So these cases in point are the most successful one. The lower product, page X2, is the most successful one. It is developed by the Narcology Department and the Almazov Center jointly in Skolkovo. And this technology has been tested and also implemented. By the time we were trying to expand this technology into psychiatry hospitals, we found out that the incidence of unfavorable reactions is lower. We may also appreciate that the frequency of polymorphism can be distributed over ethnic groups. We know that Russia has lots of ethnicities, and therefore it is important to take into account these mechanisms when we develop personalization mechanism. Our genetics, the group by Professor Balanovsky, allow us to develop a pharmacogenetic atlas of the Russian Federation. On top, you can see the frequency polymorphism correlating with the frequency and sensitivity to warfarin. And you can see that the color becomes more intense from West to Europe. This is because Russian patients and the patients in Russian Yakutia need different doses. The Yakutia needing 0.5 tablet instead of 1 full tablet for St. Petersburg. Whereas the lower graph is protective polymorphism, which is normally low if the patient is intaking dabigatran. Look at this scenario. It seems the nature knew that we are going to have such drugs. And this is highly crucial for further implementation of personalization mechanisms in Russian Federation. What else is to be done? We should suggest clinical validation of this approach. And on top of that, we must validate the clinical feasibility of this project. Nothing can be implemented if the clinical feasibility is not assessed. To this end, we have also developed a specific design. We have published a smaller guide in the Roszdravnadzor newsletter, and we are jointly collaborating with this monitoring body. Regrettably, this research is poorly financed, and it is normally financed by way of grant support. Therefore, our task is to reinforce our meta-analysis and systematic reviews. In the bottom, you may see two meta-analyses, the first one for warfarin and the latter one for dabigatran and its pharmacokinetics. 2 years ago, a review was published for all Russian pharmacokinetics research and trials. Just to have this information at hand, pharmacokinetic modeling according to genetic tracing to select the dabigatran dose choice is the first pharmacokinetic pharmacological model that considers the frequency of these polymorphisms across different ethnical groups. And this demonstrates that personalization is feasible only provided that the population density is not over 20%. It is important also to implement real technologies into real clinical practice. Therefore, we must incorporate these technologies into clinical recommendations. Therefore, based on the Article 37 of the law on healthcare, it is only based on official clinical recommendations that doctors can modify their approaches. So we are happy to share our experience, and we are happy to see that it is also international experts who also refer to our research. Here you may see a reference to our works by the American Consortium, and this is a research on the proton pump inhibitor. And on top of that, how do we teach students? What competences should we shape? Personalized medicine and personalized choice of drugs is one of the competencies that a doctor must know. And based on this standard, we implement our technologies via education, educational events. We implement these technologies into continuous education system. On top of that, we also need commercialization of such technologies. This is one of my last slides. Currently, we are intensively into it with Medipal Tech and in vitro company. We develop specific sets for PCR tests, as well as tests for oncology, cardiology, and infections. We also plan to implement these technologies in the near future into practice on the basis of in vitro company, who are going to offer doctors a decision support system. Once again, as for the treatment of psychiatric patients and oncology patients, Professor Menitov, by the way, is going to talk about oncology. He's one of the top experts. We have already started to use these technologies. To this end, we use high-precision technologies, and we use pharmacogenetics, pharmacotranscriptomics, pharmacometabolomics in this respect. We must remember that none of the points in this implementation model can't be missed or overlooked. Otherwise, this technology is going to be just a dead body, and it will never achieve or reach clinical practice. When I was a student, do not think in two cases. First, when you have nothing to think about, not because your neural activity is disrupted, but just because when you have a lot to think about. And my teacher also advised me not to think when there's nothing to think on. Thank you very much for your attention. Thank you, Dmitry. Thank you for such a detailed presentations and such an insight into molecular aspects of what we call personalized medicine and personalized pharmacology today. It is not by occasion that the next speaker is going to speak about innovative pharmaceuticals. So today we had a dedicated session on gene therapy just before this session. And now I would like to give the floor to another representative of our translational medicine cluster, Alexander Garabaggio. Innovative Pharmaceuticals, St. Petersburg. Distinguished co-chairs, distinguished colleagues, good afternoon. It's a great honor for me to make a presentation for doctors, because normally I do not discuss these things with doctors. And it's a great honor for me because what I do as a chemist and biologist, as a biochemist, is first provided to you, and then you provide our results actually to patients. Before I start my presentations, I would like to extend my gratitude to Alexandra Conradi. I was in this center a year ago when just 1 week before the new year, I had to do some treatment here, but nothing serious. Okay, so I'm going to talk about the translational medicine cluster, target-guided drugs. I would like to refer to two molecular genetics experts and molecular biologists. I'm going to refer to them and I'm going to show their Hirsch factor. So 40 years ago, a prominent pharmacologist, German and Dutch pharmacologist, Hugo Kubini, started his presentations with this painting. Looking for or developing drugs is like looking for a needle in a hump of hay. So let's look at the world investment into R&D. And here are the top largest pharmaceutical companies in the world. So the best of them are those who provided us with most successful vaccines. What does it look like to develop a drug? I'm going to look at this process as a chemist, but then you are going to look at it as doctors. Normally, we look through roughly 1 million of chemical substances, then we screen the focused libraries, then we do optimization of hits, then we do the optimization of leaders, then preclinical trials, then candidate drugs, then first stage, second phase, third phase clinical trials. And only 50% out of the initial 1 million actually survive all the way through to the clinics. One particular drug, for its development, requires 10 to 15 years and no less than $1 billion just for one drug, for the development of one drug. This is an exciting comparison. This institute develops drugs against dementia and Alzheimer. From 2004 to 2018, over 500 clinical trials were performed using over 70 drugs, and none of them actually was commercialized. You can see the drugs registered by FDA. Here you can see the group of chemically synthesized and biologically or genomically, proteomically synthesized drugs, over There is no more than 30 to 40 drugs a year. And what are the reasons? Very stringent therapeutical standards, stringent requirements to efficiency and safety.
Thus, we have a deficit of blockbusters. Are the chemists to blame? On this side, you can see the presentation by one of the youngest academicians of the Russian Academy of Sciences, Valentina Namnikov. We counted at the Congress what do chemists provide. Total chemical space is commonly accumulated. It is about 100 million compounds now. So that means that each day chemists get at least 12,000 new compounds. Where do they disappear? Because most of them are considered as biologically active. And these are data on our healthcare and Food and Drug Administration. How medical drugs created? The golden age started when the hypothesis of a target was formulated. The first targets appeared in the 20s or 30s of the previous century, and the preceding works by Paul Ehrlich and John Langan. Classification of the targets, most of them are receptors. One half of that amount are enzymes and then hormone factors, hormones and factors. These are the biological structures to which the work of the medical chemists, molecular biologists, and molecular genetics will be focusing briefly about the structuring and strategy and search of drugs. The simplest and the most common structures when we know the structure of the target and the structure of the ligand, and I will show what do we mean when the structure of the target is known. It is sometimes concealed the structure-based design. Then, when we know the target, but we don't understand what substances inhibit or promote the target. This is de novo design. And finally, a very common trend in the 60s or 70s, Kussar, when the structure of the target is not known, we know the structure of the ligand. Multiple studies on cell lines, on mice, made it possible to speak about the ligand structure. And finally, a very common, similar to a hurricane research of combinatory chemistry and high throughput screening when we don't know either the target structure or the ligand structure. This is the scheme of the search of drugs. It is organized at our laboratories in this way. This is the choice of the disease and the choice of the target, primary collection of the compounds, selection of the compounds, and then I will show the slides of the incision software problems of modeling and doping. And then primary tests on cell lines. And finally, the stage of optimization. And then we reach clinics and then clinical trials. Briefly about the problems of modeling and the possibilities given by modern combinatory chemistry and computer chemistry. These are technologies of molecular modeling that allow conducting a quantity analysis of the affinity of ligands and protein substrates. That is a very useful and good information, but this is the first stage. The next stage is modeling of spatial structure of proteins. A very complicated problem, but modern computer software makes it possible to calculate these most complicated systems, particularly take into account multiple freedom degrees in proteins and the presence of the first, second, and third stage of complication. And finally, that is virtual screening of libraries. Here you can see high productivity screening and primary processing of libraries with certain limitations. Fine. the technique molecular dynamics of protein structures, which allow calculating ligand protein complexes using the methods of quantum mechanics and quantum chemistry. And these are the tools that are always improved. They're available to us. This is a possibility to conduct very long-term tests and computations that make it possible to make calculations and to use computation in future work. Our first leading scientist, Gennaro Milena, look at his here. He's a specialist in 7 in the family of proteins, P63, P63, P73 regulators of the cell cycle. And the second leading scientist, Martin Shimus, she specializes in cascade and anti-inflammatory cytokines. The first stage of our work was related with the protein P53. When we started the work, it appeared that the data in PubMed and the data characterizing the structure of proteins are not far from being complete. Due to our colleagues from Italy first, we built that complicated protein P53 by computer, and they could crystallize it using X-ray diffraction analysis, and they could corroborate that complex structure. This is a scheme that shows or discloses extensive possibilities of transcription activity of P53 protein. We can see cell arrest or arrest of the cell cycle, apoptosis, and inhibition of angiogenesis, and reparation of DNA, and a whole number of other problems that are interesting. But simplifying the scheme, we have shown that protein P53, that is a typical oncosuppressor, in normal condition of the organism is in low concentration. However, in case of development of the oncological process, its concentration increases abruptly, and its main function is to trigger apoptosis of oncogenes. However, in the organism, P53 has very serious enemies. These are ubiquitin ligases. The first stage is protein MDM2, finding P53, it marks it, and after that P53 is triggered to proteasome and then is dissolved to amino acids and peptides. That at this stage, we should find the site of binding between P53 and that MDM2 killer protein. We have found it, we have deciphered it. Thus, we had to find such chemical compounds which would replace protein MDM2 on the site of that interaction, and we managed it. I have already shown this cycle. We approximately had the first stage from 50,000 compounds. In fact, we studied 1.5 million compounds. At last, we had 10 compounds using the strategy. And I will show some of our outcomes. Everything is being patented, so I won't disclose that information now. What is shown in yellow color is the increasing activity of the compounds versus the golden standard nuclein, the only drug that has passed all the stages of clinical trials and is used in clinical practice. The second stage of Martin Shimus, inflammatory processes. We know that hyperreproduction of interleukins results in such pathologies that are shown here. And we were dealing, in addition to rheumatoid arthritis, with psoriasis. From 0.5 to 8% of the population have it, about 125, 130 million for its treatment. They mainly use topical drugs, but lately hormonal drugs were also used, and the chemist was said, "Why we don't need those molecules? We use hormonal drugs, and we close that system, and you are not needed to us." Half a year passed, and doctors came to us and said that hormonal drugs result in side effects, which even such as TB, rheumatoid arthritis, and then other diseases. So they again came to us, and in this scheme, I will show where we helped them. A non-active form of interleukin-36 is triggered due to proteasomes and proteolysis reaction. They are converted into the active form. Their activity increases 10,000-fold. Passing into the active form, they trigger serotonin macrophages, dendritic cells, and there is a process of hyperinflammation. We managed, and in this slide I show the pioneer work that we done. We managed to show that interleukins alpha, beta, and gamma promote pair by pair proteases, cathepsin G, elastase, and other. That was so unexpected that even one of the managers of that development, Marty Shimos, being one of the editors of EBSBIA for 2 years, we couldn't publish this work. People didn't believe that this could really happen, but we confirmed it by multiple studies. Then we conducted a sequence, and we determined the binding sites of proteasomes, proteases with each of the version of interleukin alpha, beta, and gamma. And our colleagues from the Military Medical Academy showed that they provoke in different way and keep the inflammation process. They showed that the process is triggered by cathepsin G, and it is maintained by elastase and cathepsin G. And following the same scheme, we manufactured chemical compounds. Much has to be done on clinical trials. So we have chosen a different trend. It is known that at the beginning of 2000 in WHO, a process of repositioning was initiated. They checked about 1,500 known drugs for which they found and verified more than 2,500 new targets. We proceeded along a similar trend, and we found the drugs which inhibit elastase. And one of the drugs which we could synthesize was a drug, Sibelistat, that is used for treatment of acute pulmonary trauma and COVID-19 acute respiratory distress syndrome. When we started that Sibelistat as an unguentum or gel with the 4th degree of psoriasis, we got wonderful results. But look, as to the further trend, preclinical trials have been conducted. We have to pass through the ethical committee, and we can also start clinical trials. How much money was saved, and a lot of time was saved. And I pass on to the second stage. This is a material provided by Mikhail Mikhailovich Gologudza. That is the innovative nanopharmaceuticals. This is actively developed at Almazov Center. These are examples of banana-size carriers that were developed by the institute headed by Mikhail Gologudza. These are nanotubes, liposomes, dendrimeres, mesoporous silica. They managed to put and to functionalize these nanoparticles a whole set of proteins, small molecules, peptides, carbohydrates, and the like. And an interesting result that was given for that presentation, enhancement of, in fact, limiting effect of adenosine during its absorption on the surface of silicon nanoparticles. I think statistically it is quite reliable and interesting. And finally, noteworthy is also the concept of Teranostics in oncology that is developed by his group, primarily combination of therapy and imaging therapy, chemical drugs of nucleic acids, proteins, peptides, and imaging is, of course, fluorophores and traces. Quantum points are also quite interesting, and they have reached, that have succeeded really. This is their work on functionalization of the core and casing that makes it possible to diagnose and treat the disease. Thank you very much for your attention. Thank you very much, Alexander Vasilyevich, for that emotional and interesting presentation. I hope that for all our students who are connected online, that was very interesting. Dear colleagues, despite quick development of personalized medicine in all spheres, from 60 to 80% of newly recorded drugs, the drugs used in oncology and generally personalized medicine, first formed due to target therapy in oncohematology and oncology, though it actively develops in other fields as well. So our next three presentations will be dealing with the issues of personalized oncology, and we have invited the leading experts in each field. And the first floor will be given to Professor Imenitov Delnamovich, who for many years have been studying personalized targets in Russia in the sphere of treatment of oncological diseases. Unfortunately, that is a recorded presentation because he couldn't attend our session today. But during epidemic, that is not always so simple. Will you please start the recording? Listen to the next presentation that is also dealing with oncology, but that is rather narrow problems studied at Almazov Center. We under the Center of Personalized Medicine, these are neuroendocrine tumors, and Elena Nikolaevna with a large team of endocrinologists and experts in radiodiagnostics have been studying this problem for many years, and they have attained very good results. Please take the floor. Mr. Chairman, distinguished Alexander Levovna, I'm an endocrinologist, but I would like to tell you that neuroendocrine tumors today are the sphere of scientific and clinical interests of a great number of doctors of different fields, and among them endocrinologists, recognizing the fact that there were a lot of advances in diagnostics and treatment of that group of diseases. I want to cover certain issues and certain problems that have not been resolved in this complicated group of diseases. This is a heterogeneous group of tumors which is produced by neuroendocrine cells that are capable of synthesis and producing biologically active molecules. In greater detail, these are heterogenic tumors that have different localization. In principle, neuroendocrine tumor can occur in any organ of the organism and in any tissue.
Neuroendocrine tumors are biologically heterogeneous, which means they are represented by different types of cells that demonstrate different regulatory mechanisms. The key feature of these tumors is that we know too few about the oncogenic drivers in these tumors. Normally, we do not know the fate of our patients. A tumor may either be a silent tumor or it may be very aggressive. So it can be low grade or very low grade or very high grade. And quite often, these tumors do not demonstrate any firm and obvious correlation, although they definitely have the same origin. So every professional association, gastroenterologists, oncologists, endocrinologists, would refer to different classifications, and we are actually united by the classification by localization, and this is how also we decide on further strategy to manage the patient. However, there is a strong wish to integrate it all. If we look at the incidence, we have most recent data for the US, because the US have a register for neuroendocrine tumors, and this register demonstrated a huge increase in the incidence from 1.03 in 1883 to skyrocketing to 8.6 to 6.98 in 2012, whereas in Great Britain the incidence is even higher, standing at 8.6 per 100,000. Today, however, we have lots of opportunities for precision visualization and visual screening of these tumors. However, there are lots of issues unsolved, including early diagnostics. So still, patients arrive and come to doctors with advanced tumors, and there are insufficient early diagnostics and early screening mechanisms. Assessing the disease status is also a challenge, and this impacts our management largely. We are missing reliable biomarkers of these tumors, and we refute to preliminary empiric approach in managing such patients. Different professional associations in different countries stay focused on the empirical approach. Empirical approach comes first, and some standard approaches come second, regrettably. Another challenge is that too few referral centers have extensive expertise in diagnostics and treatment of neuroendocrine tumors. As for diagnostics, well, today, 20 to 30% of tumors are diagnosed at the metastatic stages. So the doctors that formulate the diagnosis are quite often morphopathologists, up to 90 cases per 100,000 of population. The carcinoid syndrome. Carcinoid syndromes of intestine or gastrum are roughly up to 30%. And we cannot diagnose such tumors unless there is liver metastasis. To compare with average life expectancy, patients who had no symptoms would live for the next 7 years. However, those who demonstrate symptoms may have life expectancy of 4 years. Provided we diagnose the carcinoid at asymptomatic stage, then will we be able to extend life expectancy if there is correct therapy applied. The most unique marker to diagnose such tumors that allows us to build forecasts as well as formulate indications for treatment, this is chromogranin A. And today, this biomarker is included into the standard for diagnosis of such tumors. Hormones may also serve as biomarkers, but roughly 30% of all neuroendocrine tumors express higher hormone levels, whereas the rest not. Why is chromogranin insufficient, although it is standard? Well, actually, this biomarker has very low specificity. Its specificity is less than 50%. If this is a pheochromocytoma, then chromogranin has high specificity. However, if this is monodyne, that is also responsible for gastric tumors, then it demonstrates high sensitivity. However, if the tumor is not in the gastric tract, then the specificity falls down, and it cannot be used. Chromogranin A cannot any longer be used for diagnostic purposes. On top of that, it is well known that many diseases, except oncological diseases, can result in elevated chromogranin, like obstructive pulmonary disease or smoking. There are lots of cases when chromogranin A is elevated. Therefore, it cannot be relied on as a uniform biomarker. On top of that, we would like to have such a biomarker that would allow us to diagnose neuroendocrine tumors at an early stage, as early as possible. We would like to have such a biomarker that could be available as a prognostic biomarker, allowing us to forecast the behavior of the tumor so that we could rely on this biomarker to choose the most optimal management strategy. And we do have such a biomarker. Well, actually, this is the unstudied issue which I'm going to focus today. Well, neuroendocrine tumor, as any other tumor, is able to produce some information into the peripheral blood. Well, in fact, it produces information on the biological behavior of the tumor, or sort of a biological portrait of a tumor that we can understand from the peripheral blood screening. This is the so-called NET test or NEO test. The NET test is based on the evaluation of several genes expression. I mean the expression of the genes in blood, the genes that are directly associated with this particular tumor. Its proliferative activity is its secretory activity and its ability to respond to therapy or inability to do so. So here you may appreciate a protocol that brings together the key steps in neuroendocrinologic tumor screening. So we obtain a blood sample, we identify the RNA, we do the PCR, the RNA PCR, and then you need to do some mathematical calculations. It's quite a complicated analysis. Ultimately, you end up with a scale to evaluate the disease activity, depending on the number of scores and the number of points or the percentage, and this is also a diagnostic test. If you look today for specific localizations, for specific sites, such analysis is already done. To compare, well, I translated it as liquid biopsy, meaning that the sample is peripheral blood sample. If we compare with regular biopsy, well, what do we base our diagnosis on? And we also need histology. So if we compare tissue biopsy, the sample is obtained intra-op or pre-op by punction. In case of NET test, NET blood test, this is a regular capillary blood test, and we do histology. And if we include immunohistochemistry, immunogenetic analysis, this allows us to understand a lot more. Here we see quite complicated interpretation, and it is called molecular genomic analysis. What else could be possible? We can do repeat blood sampling if the tumor starts to progress. But anyway, you need a substrate. So obtaining a repeat sample is surgical biopsy. In case of obtaining a blood test, this is any blood test obtained in real time. Well, invasive surgical procedures have actually higher validity than blood tests. Well, interpretation, someone may argue, but morphological assessment is always relative. Well, in this table, you could see subjective. Well, I decided to reformulate it into relatively subjective, because you can't say it's 100% subjective. NET tests are believed to produce an objective, unbiased mathematical algorithm, allowing us to monitor the disease. However, monitoring requires regular blood samples. Here, we obtain the real-time assessment. Let's look at the number of publications from 2015 until now. PubMed, Google, and you can see the number of publications has skyrocketed. So actually, those who are very much into neuroendocrine tumors are really excited about this topic and this new test. And once again, if we compare this test with neochromogranin test, well, look, all these neuroendocrine tumors. In the center, we can see 97%. The 45% is when the test works. And chromogranin is 45% only. The same stands true for other localizations. In the smaller intestine, this test looks well. In the pancreas, it fails, and it is even worse for lungs. So the chromogranin test can be a diagnostic test, but a lot more than that. Now, diagnostics. The majority of papers that are published now refer to gastropancreatic tumors. However, there are cases looking into lung tumors or paragangliomas or pheochromocytomas. So I'm going to focus on gastroendopancreatic tumors and lung tumors. So the SKO, so we know that the NET test is more reliable in terms of average values in gastroendopancreatic tumors, and if the disease is subclinical. So those who have the diagnosis of neuroendocrine tumors and those who have not yet been diagnosed with as subneuroendocrine tumors. Well, there is a minor deviation, there is a minor contrast, but we see the same for metastasis, I mean distant metastasis. 40 versus 26. And we have valuable and relevant differences. The bronchopulmonary localization, well, such cases are rare, and I also referred to this particular paper, and the authors even were granted with some kind of a prize for this paper. Well, of course, it is all relative. It is clear that the NET test is less than 20 or 20, which means that they don't have neuroendocrine tumors. But those who had neuroendocrine tumors of bronchopulmonary localization, the average would demonstrate different average values. The specificity is highly high for this test. Those are diagnostic tests. As for the tumor status assessment, whether it is a stable tumor or a progressing tumor, you can see very well on these graphs that progressive disease demonstrates normally higher. It demonstrates higher risks if it is a progressive disease. Let's look at the red line, and let's look at the specificity and sensitivity of the NET test. Both of these parameters are never 100%, of course, but this would be a mistake if we had 100%. Therefore, both tests just demonstrate high specificity and sensitivity, and thus a high prognostic value to prognose the stage of the disease and the status of the disease. On top of that, another exciting point is the data referred to and demonstrated by the authors. They do patient profiling for the NET test, and those who were were clean from any symptoms, those who were asymptomatic, But you can see those in red circles, whereas the triangles demonstrate and correspond to progressive disease with high specificity. So the test allowed us to discern between a stable disease, asymptomatic disease, and unstable progressive disease.
If we want to predict a response to therapy with radionuclides, peptide therapy, just look at the results of tests prior to after treatment. There was a surgery and a therapy with litchicin 177 prior to treatment. And after treatment, there is a difference. And after treatment, it is much lower than before treatment. Now, this test can be used for selecting or prediction of the response to peptide receptor radionuclide therapy, whether the tumor would respond or not respond, as compared to classical test of chromogranin. This difference is not to the benefit of chromogranin A. The test showed very high specificity and sensitivity. Generally, if we answer all the questions that are answered, probably one net test. Probably I'm taken away with that net test, but we have discussed the possibility of working to initiate this work in our country, but we need mathematicians. We won't be able to work without mathematicians. We can diagnose, we can conduct monitoring of the tumor, and we can assess the prognosis. And I hope that soon we'll have peptide receptor radionuclide therapy. That is our dream. And we're able to select the patients who would be candidates for the therapy. Before I finish my presentation, I would like to tell you that it is good if we have that test, and we'll have that test, because many things depend on the place where the assistance is provided to patients with neuroendocrine tumors. I would like to show how the scope of the medical assistance and the survivability of the patients depend. As to the extent of volume of the medical care, I mean not only the amount of treatment and the patients, but the possibilities of the center. If the center provides a lot of care, the survival rate will be much higher, and I think that our center has such a possibility. Everyone is speaking about his or her center. Now we are recognized by the European community as the center of excellence on neuroendocrine tumors, on pituitary tumors. And I hope that would make it possible for us to jointly develop such a test and to provide early diagnosis of the tumors, to treat better and to increase the survivability of this group of patients. Thank you. Thank you very much, Elena Nikolaevna. We have good news. Everything is okay with the presentation by Evgeny Novich that will be started now. I won't announce it again. Dear colleagues, my apologies. In half an hour, I am going to start my conference, so I have to open it. So I wish you a successful final stage of the conference. My apologies, but I have to leave. Andrei, won't you understand it? Because we're a bit late, so thank you for being with us. Thank you very much once again. Take care of yourself, as well we should do now, and good success stories to you. Technical service, what is the situation with Eminetev's presentation? Good afternoon. Per se, the term personalized medicine in oncology it might seem strange, because undoubtedly, when we're speaking about any medical event, it has always targeted the patient, and by definition, it is personalized. But in my view, the triumph of personalized medicine is related with surgery. Just see, in the same place, experts come together, different experts come together, and it is obvious that any surgery is really the action of a personal nature. In any way, when we are talking about the general application of that term, we mainly mean some specific features of the patient that make it possible to adapt certain medical events to this patient, to adapt them, take into account certain passing away from the average standard norm. This might be the analysis of the pathological focus, if that is a tumor, the analysis of the patient-specific features, for instance, features of metabolism, or certain drugs. And in modern interpretation, instead of the term personalized medicine, we often use the term precision medicine, because we do not mean the analysis per se. For instance, the bacteria on the cultural medium from the infection focus, that is also an individual laboratory test, and antibiotics are individually administered based on the sensitivity range. When we mean the modern interpretation of the term, these are mostly omics studies, those technologies that are related with DNA, RNA analysis of genome and proteome. In fact, personalized medicine and oncology, it seems to me, is dated with the description about the first series of oncological patients. The first series of oncological patients refers to memory breast cancer. It is dated about 2000 of 1,500, 1,000 years BC. The doctor from Egypt had 8 patients, and he recommended, if there is only a breast cancer, it is recommended to use hot iron. That type of ablation would be based on surgical intervention. Now, if the tumor propagated to the axillary area, so lymph nodes are affected, then the disease is a systemic one, and then the treatment would be impossible, and the patient would have an adverse outcome. But this is personalized medicine. If we take the basis of modern oncology of solid tumors, the key question whether we need surgical intervention or is it impossible and we can have only systemic treatment. In some cases, they offer the combination of the methods. Molecular diagnosis in oncology. There are some 2 areas that are under the standards. I mean, the science on inherited tumoral syndromes, that is for choice of therapy, and some things are at the experimental phase. It is known that a tumor, since it grows under unfavorable conditions, sometimes it is disintegrated, and thus mutated DNA goes to the blood flow. A lot of methodologies are related with detection of mutations characteristic of the tumor, and on this basis, some measures are taken on planning of treatment and on a midterm basis that will become the basic component for early diagnosis. Another feature today is differential diagnosis of the tumor. When we can see metastasis, we don't know where it has come from. Differential diagnosis is made by clinicians, and morphologists try to reveal specific markers in the tissue. Molecular biology can offer a lot of things because the tumor has certain tissues, specific mutations in tissues, inherited tumoral syndromes, one of the most important areas of modern oncology. There are there is a wrong notion that inherited cancers are a rare disease. There are different, family trees that comprise a lot of oncological diseases are quite rare, but many inherited cancers appear only in women, so that those family trees cannot be extensive. But in fact, if we take the disease rate of inherited cancers, it is quite great. One of the, for instance, ovarian cancer, each fifth case of ovarian carcinoma is an inherited diseases. When we have predisposition to ovarian cancer or breast cancer, this is an indicator for breast cancer. Each 10th, the 12th tumor is related with inherited defect. Those who work with oncological patients have a lot of patients with inherited tumors. Some ethnical aspects that have to be taken into account. So just think why people are divided into nations. Each nation has its progenitors who appeared to be successful, and they could give the rise to a whole ethnical group. By definition, each tribe, each nation has a unique pool of inherited mutations. That's why, if we are talking about genetic diagnostics of inherited diseases, we shouldn't copy what has been done in Western Europe and the USA to all other races and nationalities. I will give an example. In 2012, we were lucky. As a result of long-term efforts, we managed to discover the new gene of inherited breast cancer. That is a BLM gene. We showed that about 1 of 2,000, 1 of 3,000 persons are bearers of mutations in the gene. That is heterozygous gene. Mother's or father's copy is disturbed, and BLM syndrome appears when that disturbed gene is inherited from mother and father. Then that is a serious disease. Immune deficiency, tumors might occur in the children, and very low life expectancy. If it is heterozygote only in one group, these people have only departure from the norm, that is a multiple increase of the height of the breast cancer. No other pathologies are recorded. Due to the fact that this mutation that is shown here is represented in our population. In our country, we should have several dozens of patients with Bloom syndrome. We called the genetics center asking whether they have such patients. No, we haven't seen them. And we could discover why it happened in 2016. We studied immune deficiencies. We took children with features of immune deficiency, and we analyzed all the genes which are known that are responsible for immune deficiency. Thus, we had multi-gene diagnosis in all symptoms with the syndrome primary immune deficiency. As a result of the diagnostics, we found the patients with Bloom syndrome. That is the mutation which we saw 4 years ago. And why the patient had the diagnosis of Bloom syndrome. Look at this child and the child with internet site. This child has obligate feature of Bloom syndrome, erythema on his face, and that boy has no erythema. Well, you say, you have discussed an unusual case when Bloom syndrome is without erythema. That is not the case that this case is unusual. That is Slavonic mutation present only in Slavonic tribes. It gives complete Bloom syndrome, immune deficiency, low height, predisposition to tumors. After our work, There were several children in Czechia with the same mutation. They also have Bloom syndrome, but they have no erythema. This is an example of the importance of the national factor. And as to diagnosis of inherited cancers, that is not so simple. There are often phenocopies when different mutations result in clinical shows. And in this situation, more and more diagnoses of inherited cancers are used on multi-gene sequences of the new generation. Today, there are several dozens of different drugs which are used for treatment of oncological diseases. The problem is that when the oncological patient starts, you choose that not on ideal characteristics, but on the basis of probabilities. So that scheme has 50% of response. So with the patients with large intestine cancer, and each patient should have his own efficient and individual therapy. In fact, in the last 10 or 15 years, there was a significant progress because there appeared a whole range of target drugs that are associated with certain mutations. And on this slide, genes are shown in which significant mutations occur in terms of treatment, and classes of drugs are presented that are administered if mutations are recorded. In fact, the work in this trend has changed the life trajectory of some oncological patients and their families. For instance, the patients with lung cancer prior to such therapy-related mutations at the stage of metastatic disease could live from 6 to 12 months. And just look, this is our own research related with the patient who had translocation of ALK gene, and there is a whole range of drugs that can be used successively. The main figure is life expectancy, 70 months, so it appears that this is about 6 years and 6 times more than we expected. Life expectancy of these patients before target therapy and personalized medicine. So personalized medicine is operational. That is a wonderful case. In 2015, we had a patient with cancer, and there were inhibitors. We made a test, and he had a mutation of Werner-Gar. We appeared that all the possibilities for drug therapy were ended, and we used the inhibitor mutated verifirab in the tumor of gastrointestinal tract. The therapy was not efficient because gastrointestinal tract tumors express receptors of hydrothermal growth factor, and therefore they showed cascade. Due to the activation of that receptor, the tumoral cells are removed from the action of the drug. Hence the decision we have to give a conservation of inhibitor verifenib panitumumab inhibitor of receptor of the growth factor. The patient who had no chance to improve the situation had a total regression of the tumor, this combination. After 1 year, I get a letter from the American doctor. You described this case, and now I have a similar patient. Can you tell me what is the duration of the response? Then we had the duration 1 year, and I wrote, as it was, the doctor thanked me. Due to your letter, he said, the insurance will cover the treatment. That is an example of personalized medicine. Another interesting case, NTCM. This is an international recommendation, which is the basis for choosing the therapy of oncological patients for planning the treatment. That single case is a part of MTCM recommendation because that is a rare disease with rare mutations. The experts of the expert council of MTCM think that one case is enough to say if you have a patient with gallbladder cancer, you have to make reamputation and you have to use combination of both inhibitors. That is also personalized medicine. One observation was enough to be within the standard. Another interesting example, about 1 million persons per year have large intestine cancer, and the disease is related with the mutation of Genera that have 60% of patients.
We do not have any RAS inhibitors today, but we can use TOR kinase inhibitors that is activated by RAS. However, the monotherapy is not effective for colorectal cancer because the tumor cells have the so-called autophagia mechanism. The autophagia inhibitors are well known. This is proquinil, which was developed for malaria and to treat inflammatory diseases, including coronavirus. This is the case of a patient with advanced colorectal cancer with no reserves for conservative therapy, and the patient is prescribed binimetinib plus hydroxychloroquine and bevacizumab. And actually, here we see an absolutely just fatal patient. Mutic associated colorectal cancer. This is the syndrome, an inherited syndrome when tumors have a high mutation rate and high immunogenicity. Metastatic colorectal carcinoma patients. This is the number of patients in 10 years, overall 6,000 patients, and they had KRAS mutations. Why do we need this? Because the KRAS mutation marker is the marker of this rare metastatic colorectal carcinoma, inherited colorectal carcinoma mutation. And you see that only 200 patients have the KRAS mutation. It is them that we test, and we find out 9 patients with the MUTI-H gene mutation. What do we want to do with the immunotherapy? Here you can see the regular positive control, microsatellites and lots of lymphocytes stained red. And this is mutic cancer with a high rate of mutations, and therefore we administer immunotherapy, and the patient is in remission for over 3 years, which is indeed fantastic, because this was a patient with no reserves for conservative therapy. And ultimately, the final slide, here you may appreciate the key dimensions of personalized oncology and the key dimensions for further research and application of personalized medicine in oncology. Thank you very much for attention. Well, we can only extend our virtual online gratitude to Professor Imenitov. Well, regrettably, we are even unable to ask questions because this is indeed a fascinating report. Distinguished colleagues, I'm really sorry that we are extending our time limit, but I must now give the floor to Professor Hellman. Professor Hellman is our long-standing partner, and our partnership dates back 20 years ago, and he's our partner. She's the partner of Institute of Hematology and Oncology, and we are really grateful to Professor Hellman that he was able to arrive to our forum in person.
CML is the poster child of targeted therapy and of personalized medicine in oncology. And the reason is that the elucidation of the molecular pathogenesis of CML led to the production of a targeted inhibitor, of the target, of the responsible target for the generation of CML. And I'll tell you, I try to be brief in how we went about it, how we tried to generate a cure of CML on the basis of this targeted therapy. Because if you have a targeted therapy, that does not mean at the same time that you can cure. This one?
Yeah, okay.
Oh, that's a microphone. Okay. Okay, you know, so I got that. So, and the instrument we have been using on a European, on an international basis was the European Leukemia Net, which was started shortly after the turn of the century, and it promotes cooperative research for curing leukemia by regular meetings, the development of common standards and diagnosis and treatment, common projects and trials, and recommendations at the highest scientific level. The European Leukemia Net developed recommendations for all leukemias and for all methods necessary for treatment and diagnosis of leukemia, and I just concentrate here on what we achieved with CML. This is the latest recommendations for treating myeloid leukemia. It's an international panel, and as you can see here that Russia is represented well on that panel with the names of Anna Turkina from Moscow and Andrei Saritsky here from St. Petersburg. And the participants in this enterprise, you know, are depicted here. It's centered in Europe, but there are also centers in the United States and also in the Far East, in Australia and around the world. And this is the end point, I would say, this targeted approach giving us the chance of a personalized treatment of CML is the result of 156 years trial and error. And I think that just has to be kept in the back of one's mind. And the current aims of treatment are survival of normal duration and at a good quality of life, and treatment-free remission. I mean, a good quality of life is when you don't have to treat. If the patient has the idea, you know, he is cured and he doesn't have to use drugs. And now, you know, is this a pointer? We don't have a pointer here. Oh, I see. You know, I do it with a little error. Yeah. So, you know, I go back, and what you can see, the survival, 10-year survival, 10-year survival is approaching 90%, which is virtually the same the general population, and you see that here. This is the absolute survival here. This is 14, 15 years, and this is the relative survival. This is close to 95%. So, I mean, we still have a few cases of CML that progress to blast crisis, but this is just a very small minority. Otherwise, we would be at 100%. So, the rational diagnostics we have to consider. I think the most important of that is that you have to do a polymerase chain reaction for detecting and typing the BCR-able transcripts. You know, this is the responsible structure for CML. And the targeted therapy is directed against this structure. So it is very important to monitor this structure, and the monitoring of this structure has become the poster child of molecular monitoring at large. All the other oncological diseases and leukemias, they look at CML as a positive example how to do it successfully. So there is also a measurement of the individual risk of a patient. Not every patient is alike. And so there are risk scores, and there is now a new risk score, and this is supposed to be taken. This is just a brief mentioning. And there have been risk scores before. The current risk scores is ALTS score. The old risk score is called the so-called score according to a name, and the new score is just better. Let me put it in a nutshell. You know, the details are not so important right here. So, but you know, the monitoring, this is a centerpiece of the molecular management and of yeah, well, this is great. Of molecular management and of treatment monitoring and individualized treatment. And you know, there are certain time points which have to be reached. The level of the molecular measurement has to reach certain amounts of molecular, certain amounts of levels. You see that on the green, the green column that shows you what level of molecular expression has to be reached by 3, 6, and 12 months, and it has to be go down by I think 3 orders of magnitude, or even more orders of magnitude. And if you do not reach these levels, we have definitions of levels that are called warning, and then we have, of course, certain levels which are not acceptable, and so they are called failure. And there are now, meanwhile, available time courses of deep molecular reactions, and I just wonder, you know, how, you know, I can, you know, this arrow that does not, no. That does not really, you know.
Doesn't work.
Yeah, I think you have a.
Okay.
You have a good hand, Dr. Conradi, how to do this, you know. So, you know.
Perfect.
Okay. Yeah. Okay. But now let's go back. You know, this is the line which I mean. You know, and if you reach a level of 4 orders, MR4, 4 orders of magnitude below the standard, or 4.5 orders of magnitude below the standard, called MR 4.5. This is called deep molecular response, and patients who reach these levels have a very low likelihood of progression, less than 1% in 10 years. So this is just something which was very positive. I've been dealing with CML for half of my life, and to realize a development like that, that's like a miracle. And so there are several options. We have choice of first-line treatment, and this is the original molecular targeted agent, imatinib. but there are also now newer ones because under imatinib, this BCR-ABL molecule develops mutations, and so new drugs, new tyrosine kinase inhibitors were developed that recognize these mutations. And with these mutations you have to choose now according to certain principles, but meanwhile all of that is very expensive, and now we have a generic imatinib which is cost effective and can be used. And of course we have to consider that these tyrosine kinase inhibitors have a specific toxicity which may be dangerous, but somehow we were so lucky. There is really, something serendipitous about imatinib because this is a very effective drug at almost no long-term, at virtually no long-term toxicity. I mean, there's minor side effects, but altogether this is just a great drug. And I mean, there are also some other options you have, but I won't go into this. Then if there is treatment failure and you have to choose a second-line treatment, there are also certain principles, which allow a personalized approach. You have first, of course, to assess the compliance of the patient. If the patient doesn't take the drug, you are at a loss, then you just can't stop. But then also one can look at the disease stage of the patient and can adapt then the treatment according to the disease stage. There are, for instance, additional chromosomal aberrations there that can be addressed, and you also can test for BCR-ABL1 kinase domain mutations, and you adapt your treatment according to that. I mean, the kinase domain mutations are much rarer than originally thought, less than 5% in 1,500 patients over 10 years, so that's not very frequent, but it has to be considered. And then if you switch to a second generation tyrosine kinase inhibitor, which occurs in about a quarter of the cases, then the switched patients have a worse prognosis. So, they are probably a kind of patients who have already genetic changes which promote the progress to an acute phase. Then we also have defined to promote personalized medicine what we do when treatment fails. In case of failure, that means just resistance, so then a change of treatment is mandatory, or in case of intolerance, some patients tolerate treatment better than others, and then of course the choice is depending on the patient and on the physician. And then in the case of warning signs, you remember the yellow column, that is in between, and then the change is optimal depending on the treatment strategy and so on. I don't want to go into too much detail in this context here. This is supposed to be a little more a general lecture, and so I just show you, there are clear definitions of mutations and what you do. This is really the background and the backbone of the personalized treatment. And the same is also with the choice of second-line treatment. You have to consider the type of side effects and the comorbidities of the patient. If the patient has also a diabetes or a coronary heart disease, then they are disqualified for certain types of tyrosine kinase inhibitors, and there are clear definitions here, comorbidities and contraindications. This is a list, and that you can read that up. I don't want to go into detail here. And this is just the basis for the most recent and most toxic tyrosine kinase inhibitor, ponatinib. You give that when everything else fails. And then also there are certain limits you should give, and there is an indication that if you give 45 milligrams, then there is a certain advantage of survival, but also a certain toxicity, and so you can adapt according to that curve the optimum concentration of punatinib. And this is an example of what you observe with some of these tyrosine kinase inhibitors. There are now 10 and more year evaluations available, and here in this case you see that this tyrosine kinase inhibitor has a 25% serious vascular toxicity after 10 years. So you just cannot use this drug so well over a longer time, and you have to try to discontinue that very, very early. So if punatinib and other TKI fail, that happens in a minority of patients, then you can transplant, and the transplantation has certain indications, like for instance tyrosine kinase inhibitor resistance to the initial second generation tyrosine kinase inhibitors, most important resistance to punatinib, or the emergence of high-risk additional chromosomal aberration. And this is just that has to be taken care. It has to be taken into consideration, and here you can see the progress over the years that has been made by the introduction of tyrosine kinase inhibition in blast crisis, those who do not respond so well. But even in blast crisis, the survival now, blast crisis in 5 to 7% of patients. Blast crisis responds better now if the treatment basis is tyrosine kinase inhibitor. Here you see the historical comparison between conventional treatment and tyrosine kinase inhibitor treated blast crisis. So there is a clear difference, and I just learned this afternoon in the session on personalized treatment in blast crisis that if you take great care and you treat these patients very well, very early, and with the right treatment modality, you can push these curves upwards. So this is now a little more on blast crisis, and you see if you treat well early, this is 30 years ago. 30 years ago, only busulfan was available. 75% of the patients got blast crisis. And now, with imatinib, 30 years later, here after 14 years, only 6% develop blast crisis. That means you can prevent blast crisis by targeted treatment in more than 90% of the cases. This is brief mentioning of the recently detected high-risk additional chromosomal aberrations, which deteriorate the prognosis of patients, and you just have to know, and you see if you have this kind of abnormality, the prognosis is much worse. They have a survival after 9-10 years of only 50% as compared to low-risk additional chromosomal aberrations, which have a survival of 90%. So then, if you transplant early in this situation, survival is better than if you transplant late. There are detailed instructions how you manage progression to blast crisis. This is also part of the structured, individualized management of critical phases of CML. So I mean, personalized medicine, this is a very differentiated approach of treatment. You really have to take many variables into consideration to make the right decision. But that is, how life is. It's always complicated. And in pregnancy, if we have pregnancy, there are also rules how to manage pregnancy, what you have to consider. Tyrosine kinase inhibitors are teratogenic and should be withheld during pregnancy. And it's also secreted in breast milk, and so breastfeeding is contraindicated. I mean, these are, but sperm quality and morphology are unchanged. So men who have CML and take TKI, they don't have to worry about fertility. I won't go into detail how do you do that. You know, it's just realize that these detailed informations are available, and we now come to the last, maybe future-oriented and most important aspect, that is treatment discontinuation. And just to tell you briefly, you can successfully discontinue treatment in about a quarter of patients. And there is discussion whether this quarter of the patient ultimately may be cured. You know, that means these patients don't get a drug, they don't get any treatment, and they are free of the disease. And this is put into perspective. There are 50% of the patients, you know, who do not reach the deep molecular response that is needed for this approach, and 50% do. And of those 50% who do reach these deep molecular responses, about 50% relapse and have to be retreated. They do well. Also here, only 10% CML-related deaths, with say here also the others, you know, they do reasonably well. 90% survival after 10, 15 years, you know, as you saw before. But these 25%, you know, they're the most interesting. Just again, imagine 30 years ago, every single patient with a CML died. It was an absolutely fatal disease. And now we have 25% of patients who may be cured. There are some benchmark times for deep molecular remission from various studies, and you know, after various times and various depths. This is all available, and so you have really a large armamentarium for a causative and personalized approach. These DMRs with imatinib, they are stable and frequent, you know, it's detailed here. And if you look at the result of these studies that show that about 50% of the patients, you know, stay relapse-free and in the remission, then these curves tend to look similar. This is with imatinib, this is with nilotinib, and this is a multi-center study with 800 patients, and they all look alike. 50% relapse and 50% stay in remission. There are some problems with that associated too. This is known, and you know, it can be treated. The tyrosine kinase inhibitor withdrawal syndrome, which can be addressed with anti-inflammatory drugs as needed. And then there are recommendations what the requirements for tyrosine kinase discontinuation are. And this is if treatment has been more than 5 years, if deep molecular remission has lasted more than 3 years at the 4 orders of magnitude levels, and if it has lasted more than 2 years at the 4.5 levels of magnitude of PCR levels, and if that was at least 2 years. And in summary, you know, the survival of CML patients of CML in most patients is similar to that of the general population. That's the take-home message. There are 4 tyrosine kinase inhibitors available for first-line therapy with different adverse events profiles, but no difference in survival. There is generic imatinib available, which is cost-effective, and you can give it to your patients, and they don't have financial toxicity, as they say it. You have a preferred risk score. You have information on the choice of second-line treatment. You have information on transplantation in blast crisis and on the role of high-risk additional chromosomal abnormalities. And then we have learned that tyrosine inhibitor discontinuation is feasible and safe, and that you can reach treatment-free remission in about 50% of patients at 2 years. It still declines a little bit, but the level here might be increased with better therapy, and perhaps better drugs and additional drugs. Most important predictors of successful treatment-free remission is the duration of treatment and the duration of a deep molecular response and the possible cure rate. Right now, we are conservative. We say it's 10 to 25% overall. Of course, the hope is more at 25. The pessimists say more 10%. And we still, that is the limitation, lifelong treatment of all others. So that is the message on personalized medicine in CML in a nutshell, and I thank you for having listened to me.
Thank you so much. It was a brilliant overview of the problem, and don't mind that there are not so many people in the audience, because online we have a lot. Yes, thank you.
Okay, thank you.
Глубоко уважаемые коллеги.
Distinguished colleagues, we have finished our session. Unfortunately, there is no time for the discussion, but I think if the audience has questions, they can be asked online, and we'll answer one of the questions, and we'll ask our lecturers to answer. I think that we witnessed very interesting presentations, and we couldn't even finish on time. Do you want to say something? Dear colleagues, dear friends, I was happy to attend the plenary session today, and I learned a lot of interesting things, particularly innovative methods related with genetics. For us, that is also some new things, so we'll use them at our high school as well. The more so that we have the Genomic Center. Thank you. And dear colleagues, this is the end of our session today. Some sessions in small halls are still continued, and tomorrow we'll continue our forum. Thank you.